So, with that, the first presenter is Steve Piotrowicz. The biography is in the book, so I won't make any further introduction. I'll set the alarm for 20 minutes and ask the presenters all to respect the time limits so that they respect the other presenters and they respect your lunch, because if you wish to continue beyond 20 minutes, then we'll delay the lunch. Okay, the floor is yours.
Thank you. Operational oceanography, it's a term that many people in my community use, but nobody can define it. What is it? It's not defined in the convention. In fact, if you look in the convention, the word operational appears only 4 times, only once in terms of operations, and then the rest in terms of seabed activities. What does it mean to me? It means 2 things. One is that the work is sustainable, the funding is sustainable. And secondly, that there is a community of users out there defined by operational requirements, like the weather forecasting system, which I've got up here. And what you see here is just parts of the observing system for the World Weather Watch. These are the radiosondes, which at one time used to be 1,200. There are only 600 now. And then you have a large number of terrestrial stations. You also have aircraft observations. But the big thing that the atmospheric community has that the ocean community does not have are satellites. And they can make up for a lot of these deficiencies in the observing system because of the satellite coverage. And here you see 3 different aspects of the satellite coverage. Now, most of the work that I'm going to show is funded by research funds, not sustained operational funds. Operational funds, this is the distribution system of the World Weather Watch for atmospheric data used operationally. This is just part of it. To us in the operational communities, if the data is not here, it's not used operationally. It can be on a website someplace, etc., but if it's not distributed via the World Weather Watch, it's of little or no use to the operational community. This is just part of it. This is the main background, and then here's just one of the regional networks. That system allows the data that is used operationally to have the same format, to be easily interpreted, and downloaded into operational models. There's no time to futz around with data getting it off a website. And just an example of how the data are used, what you see here in the upper right, in the upper left, is a profile, temperature profile of the atmosphere, of the ocean data that goes into, over a 1-month period, into an operational model. You see the little crosses here, those are Argo profiles. We'll talk more about those later, and they are constant pretty much in there. And then you see these ship tracks right here, and we'll go into more on that. And then on the bottom right, you see sea surface temperature, which is only done by buoys that drift on the surface of the ocean. There's not vertical profiles, and again, you see the shipboard operations. So these are the kinds of people that use data for operational purposes. What do they use it for? Here's an operational output of the US Navy model using those data that I just showed. This happens to be off the coast of Asia, plus remote sensing data. That's how that organization uses it. Another aspect of what we do in observations is to address some of the societal problems. And one of the big ones, you heard about it several times today already, is the ongoing warming of the Earth. 93, you heard the number 93%. Of the ocean, excess heat is going into the oceans. About 75% of it goes into the top 2,000 meters. 25% goes into the bottom 2,000 meters to the bottom. Average depth of the ocean is about 4,000 meters. However, the volume, about 1/2 the volume, is in the top 2,000. One of the interesting things that's happening right now is that the upper ocean that we see here, to try to balance this excess heat, has stopped warming. It's been a shock to people. I'm talking 0 to 700 meters there. But we know where it's going. It's happening, it's warming in the extreme southern hemisphere. In other words, depths below 2,000 meters and south of 40 degrees south. We don't know why that's happening, but there's a huge buildup of heat there. So even though you're not seeing it in the upper ocean, 0,700 meters, we are seeing it there. Now we've got to find out how we're going to get there. Now, I'm not going to talk about this since we've already talked about it. I'll talk about it a little bit later. Another aspect of ocean observations is the ocean acidification problem. I'll just point out here that bicarbonate, PCO2, and carbonate are three very difficult measurements to make right now. Historically, operational oceanography was basically, well, I shouldn't say operational, oceanography was done in an expeditionary mode. And some of those modes, some of those tools are just bottles hanging from wires that you see here. This little device, the mechanical bathythermograph 1938, is a very, very important device. Up until about 2 years ago, this was the largest source of temperature profile data in the World Ocean Database. By far, almost 3 million profiles. It only goes to 300 meters, and it's only temperature. To understand ocean circulation, you also have to have salinity, because 2 of the 3 types of drivers for ocean circulation are dependent on the density of seawater. The only one that's not is wind-driven. The other 2 require density, which is a function of temperature and salinity. But this device right here was actually a major factor in winning the war in the Atlantic, because it found where submarines can hide. Velocity of sound is a function of temperature in seawater. And so if you know the temperature structure, you can correct your observations to figure out where that signal is in fact coming from. However, when you use that device, there's actually a letter, the inventor of this, from Winston Churchill about it. However, when you use that device, you're very vulnerable. So, there we go. So, in the late 60s, the US Navy paid for the development of this device, which basically does the same thing as that, gives you a temperature profile, but that gives it as a function of depth. But this doesn't require a ship to slow down to use it. And you can just deploy it. It's called an expendable bathythermograph. Well, the good old scientists said, oh, this is a great tool. We're going to use it. This was designed to find submarines, not to be applied for research, not to be applied to understand the climate problem. But people have been using this, and you've seen decades of papers using that data from that mechanical bathythermograph and this. The problem with this is it doesn't measure depth. It calculates it. And there have been problems with that calculation. So again, we've been using tools to extrapolate to climate scale phenomena that were not designed for climate processes. So what was designed were these deep ocean cruises involving ships, large research ships with, we call these CTDs, bottles located lower to the bottom of the ocean. These are extremely expensive. There was an experiment, a program in the mid-90s that collected about 10,000 profiles. You see the lines here. It cost about 7,500 pounds per profile. Just per profile. Cost of ship time is the major driver there, but also the technology is very, very expensive. And this one line right here, let's see, there we go, right here in the southern hemisphere called A10 took 49 days at about $45,000 a day US to transit. Now these various lines that you see here in the lower right by the Meteor expedition, those took 31 days and 49 days in 1993 to occur. We do these cruises on 10 to 15-year cycles. So you're only getting a snapshot of it. Again, how can you do operational processes with getting these research snapshots of the data? This is, I pointed out this device right here, the XBT, and how it's been used, and little reference to the US Navy here, find submarines, that's why it was invented. I happen to have been on board that ship when that picture was taken in 1969. We were just testing it out. Now what we've done is we've equipped volunteer ships, merchant ships, with devices that deploy these XBTs And also we use underway ships to collect sea surface temperature observations and collect carbon samples, because it requires a large laboratory setup to collect and analyze carbon samples. But you see this line up here in the top here, these many black lines. They're no longer occupied. Basically, the price of these, the cost of conducting these observations, this is not very expensive, only about $50. And the availability of ships has been declining, and in the last 20 years, we've dropped by a factor of 2 in the number of devices we deploy for our operational system. So we've got to find another way. Just further going along the lines of that is on the usage of it. These are what we call surface drifting buoys. They collect only sea surface temperature. They can't collect salt, but you have biofouling issues there. And this is the distribution of them as of 2 weeks ago, about 1,400 out. They're only sea surface temperature, and they're primarily used for marine forecasting. In fact, a major program, it's one of the few that's really operationally funded, is in the North Sea here called ESurfMar, European Surface Marine Forecasting System. That is an operational program. But we have no control over where these go, and in fact, they will tend to clump. But how are the data used? To the right here, what you see here are data from these ships that I showed you collecting underway observations and drifting buoys. Then you see below in the middle panel the infrared satellite, nighttime satellite of sea surface temperature. And those two sets of data are combined into the bottom panel, which is the operational product that is assimilated into numerical weather prediction models. So the combination of the in situ data and the remote sensing data are used. The in situ calibrates the remote sensing are used for operational purposes. Other systems that are research/operational, you've heard of the tsunami warning system right here, the dart buoys here, and this is the distribution of them. That is an operational system, and in fact, it is funded by operational funds. That is an operational system. The Banda Aceh activity event really triggered that. However, there are also, you see here these moorings, which are tropics, e.g. El Nino, for El Nino prediction. You see these lines across the tropical Pacific here, the vertical lines. Those in the Pacific now are funded by operational funds. They used to be funded by research funds. However, the problem is, again, the ship time. The cost of ships to service those. In fact, about a year and a half ago, we had a crisis where much of the array in the Eastern Pacific was down because we couldn't get a ship there. There are also research applications here, and I've got those moorings here over on the right, and these are scattered around the globe, but those are used, the data are made available, but those are used for research purposes and funded for research funds and can come out within a 1 or 2 year notice. But again, these buoy moorings require large ships, very expensive. And getting to the ships conducting those long lines I showed you, okay, once every 10 or 15 years you get back. Well, what you have here are two animations of the 97-98 El Nino event and the partial event in 2015-16, where you see the evidence of the evolution of the warm tongue in the 97-98 event, and then the cold El Nino, La Nina event coming across, whereas in 2015-16, you do see the somewhat of an El Nino, but the cold event didn't come all the way across. That's why people are still wondering what that was, and they're still trying to figure that out. But the issue here is that the oceans can change in a matter of months, and yet you have ships going once every 10 or 15 years. So in about 2000, the community came up with the profiling float program called Argo, and it has no relationship to the movie. It actually refers to the satellite system that it was meant to validate called JASON, which is an acronym in of itself, and then Greek mythology. These instruments are designed to last 4 to 5 years, and some of them last longer than that. They provide high-quality observations of the upper 2,000 meters of the oceans. The instruments sink, when they're deployed, they sink to 1,000 meters and park there, stay there for 9 days, and then go down to 2,000 meters, come up and transmit their data via satellites. This is just a schematic of the device right here. This has been progressing since 2000. It really started, but reached a global coverage in 2007 and reached 1 million profiles in 2012. Now 1.7 million profiles in the World Ocean Database from just these instruments. The original design of the array was a 3x3 grid, 3x3 degrees. Now that at the equator is equivalent to the area about the size of Bulgaria. So what you've got here is one instrument in an area that size at the equator. Now it changes as you go up, move away from the equator. However, ocean physics also says we're changing. Oh, good. Good time. Ocean physics actually says you need more instruments up here, and that's in fact what happens. The other thing is that it was not going to be deployed in marginal seas because the telecommunication system that we had at the time had required the floats to be at the surface for 12 to 18 hours where surface currents could drift them ashore. And the third aspect of it is 60 north to 60 south, not in ice zones, in ice-free regions only. Now, we can do ice-free in the Antarctic right now. It cannot be done in the Arctic. It's only done in the Baffin Bay right now. And if we can talk about why that's a problem. But in the Antarctic, it is being done now. And the Argo program, the International Argo program, will not deploy in exclusive economic zones without permission. Will not. And the other requirement is, of course, that the data be free and openly available via the GTS. And that's what you see is at the end of March, the number of instruments, and you're scattered all over. And there we go. And those are the distribution of instruments inside and outside of the high seas at the same time. We have a procedure for dealing with the issue of the Argo floats for temperature, salinity, and pressure to approaching EEZs. And if a country wants to have be notified of a float approaching its exclusive economic zone, they get notified by the float provider. This is an example of the message that I would get for it, and then I would send information to the coastal state. This is the information that's actually required by the IOC right in here. And this is for temperature, salinity, and pressure only. Argo is, what you see here is just the impact of Argo, and I'll get to one more slide very shortly here. In the upper right is the number of profiles below 1,000 meters, temperature and salinity below 1,000 meters, the density of it. And the median here in the upper left, excuse me, is about 27 profiles. If you look at the entire world ocean database, going back to 1870, you see the same data in the lower left, and the median number there is 2. So over a century of observations, and only 2 observations in a 1 by 1 degree square. That's an example of the information of what Argo has done. And here's somebody talking this morning about the southern hemisphere. In the top figure here is all the TNS profiles below 1,000 meters in August, the austral winter, and in the entire 50 years, and in just 5 years of Argo data in the panel below. Again, Argo doesn't care about weather. It does care about a little bit about ice, but not weather. And just finishing up on the data side of it is these are these various sources, bathythermographs, that CTD I showed you. These are the bottles hung off wires, and this is that mechanical bathythermograph. All of these are declining very rapidly here, and this is Argo floats, and it's up to 170,000. Now we've got, okay, can you play the movie? There's a little 2-minute movie here about deploying an Argo float, and it's got audio. Little 2.5 minute.
Firstly, the floats are shipped in cardboard boxes and plastic bags. The reason they're in a plastic bag is just to keep them dry while they're shipped. So the first thing we have to do is take the plastic bag off. Underneath that, you can see it's in a cardboard box. These floats normally get deployed in a cardboard box, but you can deploy them out of the box. This is one we took out of the box because the conditions were so good, and we deployed it in the same manner that we did the deep Argo float, again because that worked so well. So basically we hooked a line through the square disc there that you can see around the float. That's a stability disc and stops the float going up and down too much in the waves while it's on the surface. Hooked a rope through that, picked the float up. Right on the top of the float there you can see the CTV and the antenna, which starts to disappear overboard. Lowered it over the side of the ship and then pulled a little piece of pipe that we were using to basically hook the rope onto the float, and that let the float go free. And the float, when it's deployed, the bladder's fully retracted, so there's no oil in the bladder and the float sinks straight away. So the float, you can just see it under the waves, but just disappearing there. To do this, the ship had slowed down to about 2 knots just to make the entry into the water nice and gentle. Now the other way we deploy the floats is in the cardboard box. This can be done from container ships when they're still underway. So when the ship's going as fast as 20 knots, which is nearly 30 kilometres an hour, we can deploy them underway. That piece on the end is the water release. It has a little dissolving tablet in it that when it hits the water, the tablet lets go and the release lets go. So the crew lower the float in its cardboard box over the back of the ship. Float lowers to the water, and as soon as that release touches the water, it lets go. And you can see it just let go there, and the box floats away. At this stage, the box has no plastic on it. We take any plastic tape off it before it goes in the water.
It takes about 20 minutes. It takes about 20 minutes for the box to dissolve. And then now where we are right now are gliders. These are controllable. Profiling floats are not. Gliders, which are basically profiling floats with wings and can be controlled. This is just a sample of it. This is new technologies. This is a figure that I was going to show in that heat content of the heat going into the southern hemisphere, which you see in these red areas right here. And we're about to address that with 6,000 meter floats. There's actually 2D models. There's a 4,000 meter built by Japan and France, and the 6,000 meter floats built by the US. These cylinders they crush at about 4,500. We've got 30 of these of this version out and 3 of these out right now. Full depth, 6,000 meters. These do not park at 1,000 meters. They park near the bottom and come up. They're going to come up every 15 days. We got 3 arrays out, and we're going to put a 4th one out in 2 years. And then the other is the biogeochemical sensors. This is a biogeochemical sensor-equipped float. You will notice here that there's no carbon on it. No carbon sensors on it, and that's because carbon sensors not ready yet. There's a development underway to develop a CO2 microfluidic technology a long way away, and this is both in terms of readiness and quality control. But you do have pH on this. You have to chlorophyll, and there is an alkalinity sensor under development. If you have pH and alkalinity, you can get the carbon cycle. And then just finally, the U.S. has a program sponsored by NSF that NOAA collaborates with. We provide half the floats to deploy 40-some-odd sensors a year in the Southern Ocean. And this is the present distribution of them. It's a program that's going to go for another 3 years under research funding, and whether we can pick it up operationally, I don't know. Thank you.
Thank you very much, Steve. Okay, the last speaker before lunch is Andrea Brep, who was introduced yesterday, so doesn't need any further introduction. And again, 20 minutes if you can.
Thank you, Michael. My presentation is coming. Thank you, Steve, for this impressive historical presentation of the ocean observation. I'll take from here to explain you a bit the Tara oceans. No, is that Tara one?
Whoops. Wrong one.
No, it's not this one. So the Tara, of course, I have to thank Jean-Louis Etienne, who is here in the room, for this incredible boat you are seeing in the image. Tara was the Antarctica before, and since now we have 25 years ago, Jean-Louis. So it's an incredible boat that can go to the Arctic. So after the first expeditions, after the boat was called Tara, we did this Arctic Drift, which was the big expedition dreamed by Jean-Louis since the beginning of this project. And then after the drift, a scientist, who is not an oceanographer, a cell biologist, Eric Carcenti, came to us to propose another adventure to study plankton, because he realized that no global planetary scale study was made with new instruments and new technology on the plankton. And so he proposed to the Tara Foundation a 3-year project to study the plankton at a global scale with a common dream to assess planetary ecosystem based on plankton. Plankton, I'll take the little plankton, it's about 95% of the life in the ocean. So it's microbiota, it's from viruses to fish larvae, but it represents the huge amount of life in the oceans, actually. So the idea was to take all these, the parameters, the physical, chemical parameters that Steve explained so well, and to add biology on all its aspects, including genomics and metagenomics, transcriptomics, bioinformatics, bioimaging. So it was an adventure. It was the first time that all these disciplines were put together. So to sample from viruses to zooplankton, a set of different equipments was put in place on the boat. It was a hard work, about 2 years of preparation to go to this expedition, to build the consortium, a big scientific consortium, to do all this sampling and the analysis. So basically, this is the Tara Ocean Consortium with, I won't pass it, but I'll leave it to the scientists that can recognize some of his colleagues. So plankton, sequencing, environmental data, imaging data, data management, bioinformatics, and modeling with excellency in almost all domains. So we had 27 labs involved in the expedition. Now we have more than 300 scientists working on these data sets. So this is the path we decided for the stations. So it's about, we are talking about 35 to 40,000 samples in the 3 years of project, including the Arctic Ocean. We said ice-free in the summer, so we crossed the Arctic in the summer in 2013 to complete this trip. So the idea is also to define the stations according to satellites image to exactly match where the plankton is blooming. So plankton blooms are an important phenomena, and it varies. Plankton, it drifts. It comes from the Greek plankton, the Greek planktos, which means drift. So with all the life that is drifting in the ocean, you can't predict it, so you need satellites and observation to determine the stations where we are going to sample, and it's permanently feed on board with this observation to get in the right place in the right time. So it is a bit the image that shows the CTD, all the equipment, HPLC, nutrient sample, carbonate. Well, I won't pass it in details. I'm not a scientist, but I'm working with this project since 8 years now. So I learned a bit. So this is the internal lab of the Tara, flow clams, cytometer, microscopes, and there is a wet lab on board for the biology samples for collecting. When the Niskin bottles are up, the biologists are the first to grab the samples, to filter, and to put it into barcodes. This is the little image to show how it works on board. It's a sailing boat, it's a small boat. At the beginning, we had some doubts about the capacity of the scientists to work properly in this small place, but it worked well. And technology is helping because it's going very fast. The micro equipments, even sequencers now are on board of Tara. We have a USB sequencer on board now. First for tests, but it works. So it's going very quick. So there's the first measures on board, live images on board, and of course it is incredible plankton images. Thanks to Christian Sardet, we published a beautiful book and made these plankton chronicles, which are available online for everybody. We also sampled some coral reefs in this mission, kind of separate study of corals. So this is the logistics. It's hard to organize everything. The samples has to be good quality samples. So the shipping to it. Okay. So this I'll try to explain a bit how is this big data pipeline and how it was put in place. So the idea is we standardized all the protocols for the collection. So all these 40,000 samples were collected with the same standards, and so we could generate, organize, and analyze in data generated for this sample. So now we have a big collection. Actually, it's the largest biological collection ever made in the ocean. About 250 billion paired DNAs. We are also identifying genes, new genes, about 40 million genes already identified. Imaging, we have 6 million images of single plankton, more than 30 terabytes of raw data, which is now starting to be analyzed. It's a big work. It will take years, decades to analyze all this data. And in always open access, we are building an access portal to facilitate access, because even if it's open data, it's very complicated to find what you want in this ocean of data. So this is this was the first paper published 5 years ago to explain the holistic approach. So the idea is really to understand on DNA observation, like we have for the human genome, for the ocean genome, to have the capacity to question this database and to put questions and to have responses consistent with DNA analysis. So it's quite new. It's coming to interact with traditional oceanographic, but we are convinced that this is a revolution for marine biology, and we have to take it very seriously for the future. So this is a holistic approach, and the idea was to understand who is living with who is harming who, and to sort out the positive and the negative correlations between the organisms. So in 2015, we published the 5 papers, so it was subject for a special edition of Science. We are very proud of it, with the cover, comments, and we had several press on this, including the cover of the New York Times, the cover of Le Monde, of La Repubblica, The Guardian, and others, because it was the first planetary scale study on plankton. This is a bit to try to explain you the high throughput sequencing. associated to high-throughput imaging and physical chemistry. So the idea is all this is connected, and the database are connected between them, so you can ask questions, and the responses come with all the parameters. So for this, there's other issue. It's data management, data storage. It's a very, very expensive issue to storage terabytes of data. So we had the support of EMBL, EBI, the Genoscope in Paris, which is very important, and all the environmental data is stored at Pangea. Viruses with IMM and EMBL are doing all the imaging work. So this is the OMR, the Ocean Microbial Reference Gene Catalog, just to give you an examples of how new is this study. After our expedition, what data collected, it represents 81% of the whole microbiome reference catalog available. So comparing with the GOS, the Craig Venter expedition, you can see that we produced more than 10 times what Craig Venter did 10 years ago a bit. So we have 10.2 terabytes of sequence only on microbial reference. So what is important after the results that we discover, the scientists observed that the catalog is close to saturation. So comment on dit en français, the curves saturate. So it shows that we are going close to understand and to catalog the ocean life, the ocean plankton. So 1,000 more times sequenced than GOS. It corresponds 23,000 times more than human genome. So it's a huge effort. Also for plankton, it saturates after some curves. It starts to saturate. So it shows that it worked well. The choice of the station worked well. The quality of samples worked well, and it's really amazing what we can do with this database in the future. This is a bit to talk about the next gen of modeling, the complexity of the ecosystem. From the community metabolome, we are going to taxa and gene analysis, and then we can go to stochastic modeling. The idea, of course, is to feed ocean modeling to understand climate change impacts, basically, but not only climate change, also other features. So, what we learn, it's in French, sorry. So, firstly, it's a big taxonomic effort, genomic and morphology. We discovered, well, we proved the determinant role of temperature in the composition of ecosystem. Temperature is the main driver that determines who lives where and who lives with who. This is very, very important in our time of climate change, accelerating impact of climate change. Also, we can say that the auto-organization of planktonic communities can be identifiable between the different realms of plankton. And this is also an important result. There's a paper published in Science on this, that the positive correlation are much more intense than the negative. It means that organism tends to live with each other, tends to go to the other, not to exclude themselves. So it's a big question for evolution of life in the ocean. And one of the questions we are trying to answer with this project is how, from single-cell organisms, how these organisms evolved, why they divided, why the single-cell organisms divided. So this is because of The scientific director of the project is one of the big cell biologists. Actually, Dr. Crescenti described the cell division, described the modern description of the cell division is from the Crescenti group in Embol. Also, it showed the importance of oceanic circulation, and also the organization of the communities is strongly correlated with the carbon flux. So it opens lots of space to studies on climate change, on the traffic chain, on carbon flows in the ocean. Some technical information for the researchers. So where are the sample collections? So the DNA samples are in the genoscope. So it's thanks to the COA and the CNRS for the support. It's about, just to give you an idea of the sequencing effort, it's about 50 people for 2 years working on the sequencing to give this database. So all the DNA samples are managed in the genoscope. Zooplankton in Villefranche, very close to here. The protists are in Roscoff, in charge of, with the leadership of Colomban de Vargas, one of the very brilliant scientists that created this project. The microbial culture are in the Bigelow, University of Maine. Some of you know Emmanuel Boss. Dr. Boss is in charge of all the microbial studies. And finally, we have also Barcelona, the Center of Science in Barcelona, which are in charge also of some aspects on the microbial collection. And I think that's all. Thank you.
Thank you very much, Andre. Well, it is 1:10. We should break now, I think, and we can resume at 2:30 sharp. 2:30 sharp for the 2 other presentations, plus some time for question and answer on this panel. So thank you very much, and bon appétit.